Good afternoon, everyone. So it's already 4:05, and we successfully kick out the previous class. So I think we should get started because we have amazing talk ahead of us. I will keep my introduction short. Professor Philip Holliger is a program leader at MRC Laboratory of Molecular Biology in Cambridge, UK. Phil graduated with distinction from Swiss Federal Institute of Technology, in other names ETH, at Switzerland. And then he came to UK, did his PhD with the Nobel laureate, Sir Gray Winter, during which he started really amazing work with nucleic acids and synthetic biology. And he did a very short postdoc there, then started his tenure track at MRC, LMB, and got his tenure in 2005. So his lab aims to better understand the foundational principles and the chemical logic that shape our genetic system, mostly DNA and RNA. I don't know, like maybe once in a while, you may wonder why we have DNA and RNA as our genetic materials, why it has to be ribose, why it has to be 4 bases, if not more or less. So to answer that questions, Philip's lab is really doing amazing work to try to recapitulate what happened initially, how the first life on Earth emerged, and why this whole replication system is possible with simpler chemical structures to begin with. Without further ado, I just want to welcome our speaker today again to deliver the talk. Thanks so much. Okay.
Thank you. Can you hear me? Thank you, Xiao, for the introduction and for inviting me here and giving me the opportunity to tell you a little bit about our work. So this being the town of Jack Szostak and David Bartel, I'm just going to talk about polymerase ribozymes and actually not talking much about the XNAs. So really, what we start with is this central dogma kind of diagram introduced in the 1970s to sort of summarize the idea that information flow in biological systems is mostly unidirectional from DNA into RNA into proteins, but then immediately people recognize there's a sort of strange dichotomy to this system in that we need nucleic acids to make proteins, but then you need proteins to make nucleic acids. So how do you get a system like that started up? How do you boot up life? And this is over 50 years ago. Francis Crick, Leslie Orgel, and Cole Vose, and also Alex Rich here at MIT sort of proposed an idea which was pretty far-fetched at the time, which was that there was a primordial biology which was much more primitive and where RNA was doing most of the sort of heavy lifting, both catalysis and information. And although that's still kind of there is no sort of concrete evidence for such a, as it became known, an RNA world ever existing, there's a lot of compelling circumstantial evidence. Maybe the smoking gun is really the structure of the ribosome, sort of central hub of protein synthesis in all cells, which is mostly RNA, including the peptidyl transferase center. And really, a cornerstone of this idea of an RNA world is this, that it must have started with maybe an RNA that gained the capacity to make copies of itself, to self-replicate. And so many people, such a RNA does not exist in nature, so many groups have been searching to build such an RNA replicase in the lab to maybe study the properties of life's first genetic system by proxy, by studying a molecular doppelganger. And we sort of joined this effort, starting from this truly amazing kind of polymerase ribozyme that was discovered, this polymerase ribozyme discovered in David Bartel's lab at the Whitehead. And it still sometimes blows my mind that such a simple molecule, this is about an RNA with about a bit over 200 nucleotides long, can carry out such a sophisticated molecular function, which is to read out the sequence of an RNA template and synthesize its complementary sequence. You can see it doing its job here. So just to just to introduce these type of gels. You're going to see a few of them. This is a primary extension gel. It's a nice semi-quantitative way of measuring polymerase activity, where you end label a primer, you let the polymerase do its job, and then you separate the products on a polyacrylamide gel. And you can see each sort of band denotes an incorporation step. So this RNA polymerase ribozyme can do about 14 incorporation steps, and then it's spent. So that's not quite ready for self-replication, but we thought our first talk should be, this ribozyme is about 200 nucleotides long. Can we engineer this further? Can we evolve it to acquire the ability to synthesize RNAs that are at least as long or ideally longer than itself? But before I start with this, there is something that always bothered me about the RNA world. I mean, I don't know how many of you have worked with RNA. It's a pretty fragile molecule. If you expose it to alkaline pH, to high temperatures, or to divalent metal ions, in particularly transition metals, or God forbid, combinations of that, it quickly degrades. Now, that's all very well in the lab, where you can control conditions in nice clean Eppendorf tubes, but on the surface of the early Earth, with sort of meteorites raining down, hot boiling springs, come on. RNA is a pretty perverse choice as a primordial genetic material. Now, other people have proposed a pre-RNA world that would then be based on a much more resilient genetic polymer, which then transitions to RNA. But it's worth saying that there's zero evidence for that. So we wondered if we could think of environments where RNA would make more sense. And on the face of it, the answer to that is pretty obvious, because what do you do both in the lab and at home if you're dealing with sort of perishable things? That's right, you put them in the freezer. But we also need the RNA to be an enzyme. And certainly protein enzymes don't take kindly to being frozen. So this is T7 RNA polymerase. You can see it happily synthesizes RNA at 37 degrees, but when you freeze it, it's dead. But the polymerase ribosome is very different. You can see not only is it active in the ice, but if you just wait a little bit long enough, it pretty much shows the same activity as at ambient temperature. So it retains its activity when frozen. And in fact, if you adjust the conditions just a little bit, it can synthesize RNAs that are actually quite a bit longer than it can do at ambient temperature. So it is a little bit like the tortoise and the hare. It's a bit slower in the ice, but it just keeps going and going and making more and more RNA. And the reason for that is, and this wasn't discovered by us, is that it would be wrong to think of ice as a sort of homogeneous solid medium, because what really happens is when you freeze an aqueous solution that doesn't just contain water, but contains RNA and maybe magnesium and some nucleotide triphosphates, the water freezes out first and excludes the solutes into a liquid brine phase, the so-called eutectic phase, which surrounds the ice crystals and actually stays liquid at sub-zero temperatures. And this is accompanied by a tremendous concentration and dehydration effect, which sort of compensates for the low temperature and accelerates the synthesis reactions. So this really looks like a nice environment for RNA to do its job. And in fact, all the reactions you see from here on are done in ice in our lab. So as a next step, we tried to do evolution in ice, and we selected a new variant of the polymerase ribozyme and to adapt it to the ice phase a bit better. And this one, I hope you can see this here. This is about 206 nucleotide synthesis band. So it can now synthesize RNAs that are longer than itself, because it's about 203. This is 206. So we're there. It can synthesize RNAs longer than itself, but can it synthesize itself? No, it can't. So that was really frustrating. So why can it synthesize these RNAs that are longer, but it can't make itself? And to figure out what's going on, we fed this ribozyme a simple pool of random RNA sequences and asked it to copy them and just see which ones can you copy, which ones you can't. And when you analyze these, these sequences, the ones that can be copied, they behave like strings, they're unfolded. But the ones that it can't copy, the moment you have a little bit of secondary structure in the RNA, this RNA polymerase ribozyme cannot read through those structures. And really this brings us to this dichotomy of RNA, that it has to be, as what Larry Gold called, it has to be both a string and a thing. And it needs to both be able to fold up and then unfold again to become a template. And really, you'll encounter this sort of theme throughout the talk. But the fact that it cannot, that this polymerase ribozyme cannot copy structured RNAs now leads us to what is an apparent paradox, because the RNA needs to fold into a three-dimensional structure to be a polymerase ribozyme, but then once folded RNAs cannot be replicated by the polymerase ribozyme. So how do you get around this? And one solution I'm going to tell you a lot about that we came up with involves that we rethink the substrates that the polymerase uses. So far, the polymerase ribozymes that I've shown you, they all use the mononucleotide triphosphates that are also used by natural RNA polymerases. But really, it was always a bit optimistic to think that at the origin of life, you'd have a nice clean pool of, NTPs kind of like ready for you. So there would be a mixture of things, maybe dinucleotides, trinucleotides, tetranucleotides, etc. And the question is, which one of these would be good substrates? So we did a number of trial experiments and eventually settled on trinucleotides, or specifically trinucleotide triphosphates, as substrates with good properties, that is, were incorporated well by the polymerase ribozyme, but they also maintain the fidelity of interaction, which you lose when you actually go to longer RNA oligomers. And just to make life a little bit easier, instead of saying trinucleotide triphosphate each time, from now I'm going to call these triplets. So this shows the problem again. So this is a sort of series of RNA templates. This is the primer. And they have an increasing tendency to fold back on itself. Now with the mononucleotide polymerase, you can see it can copy the first one, but then it's pretty much stumped, kind of like with each of the others. But if we now switch to trinucleotide triphosphates, again, to triplet substrates, you can see we can copy through all of them. And what's even better, when you sort of plot the curve of the amount of RNA you synthesize as a function of triplet concentration on the unstructured template, you get a sort of saturation curve, which is what you expect of an enzyme as you increase the substrate concentration. But if we do this on the structured templates, we get a sigmoidal curve. You just get a sort of transition at about 10 micromolar triplet concentration, where suddenly kind of like synthesis increases kind of to a much greater extent. And our interpretation of this is that actually, that it's the triplets that invade the secondary structure. So there's a sort of ratchet-like progress of synthesis where the triplets at a certain concentration become able to invade the secondary structure, leading to a cooperative unfolding of the template, which then can be copied. So at this point, we became really interested in evolving this as a triplet polymerase ribozyme, and this is our result, which we came up with. We didn't ask for it. The enzyme became a heterodimer, a mutualistic heterodimer of an active and an inactive subunit. We've recently managed to solve the cryo-EM structure of the APO enzyme. And you can see here, there's the catalytic subunit in red. And in blue, you see the non-catalytic subunit, which actually derives from the catalytic subunit, but in just kind of, a few mutations kind of completely rearranges the structure and sort of provides a scaffold to hold the catalytic subunit in place. As you can see, this polymerase ribozyme, what we from now on going to call the triplet polymerase ribozyme, has substantial triplet and sort of oligomer polymerase activity. And it can really synthesize some pretty tricky substrates. So this is the broccoli RNA aptamer, which binds this chromophore, become fluorescent. It's a sort of pretty much stay all together, a hairpin structure all the way. But now, the triplet polymerase ribozyme can synthesize them, and the broccoli is active, because if you add the chromophore to the gel, you can see this is made with T7 RNA polymerase. This is made with a ribozyme. You get a sort of an extra triplet added to the end, but they are nicely fluorescent and active. And what's better, you can do both strands, so you can actually replicate kind of both strands of this broccoli aptamer. And just to show you again what sort of structures look like, this is the structure of the related chili fluorescent aptamer. And you can see this is really a hairpin structure all the way with a sort of interrupted G quadruplex in the middle, which binds the chromophore. I don't think you could come up with a much harder substrate to replicate. What's amazing is that the triplets now also unlock kind of replication modes which are not available in nature, because all RNA replication in nature proceeds in the 5' to 3' direction. But because the triplets bind to the template, there's no reason why they can't go in the reverse, provided you give it a primer which is phosphorylated at the 3' end. And indeed, that works. So this is the forward reaction. You give it a normal primer, you give it the triplets, happily synthesizes RNA. Now we give it the reverse primer, phosphorylated at the 5' end, and you can see it does the same reaction. And what's better, if you give it both primers, it can do both reactions at the same time. Because the triplets bind to the RNA template, they can now act as primers themselves. So you can see in lane 2, this is the primer extension reaction with a primer. And in lane 4, where we give it no primer at all, just triplets, and you can see you can make exactly the same product. And you can use that synthesized product and prime it again, just using triplets, and again, make both strands of the RNA. Now, initiation from triplets has a number of consequences. And one of them is that there's now a large combinatorial number of pathways by which the ribozyme can make the full length. So this is a 24-mer RNA template. Okay. This is a 24-mer RNA template, and these are all the potential pathways. Now, if you had the normal 5' to 3' synthesis, you would expect a sort of a clockwise arc to appear. If you had the 3' to 5' direction, you would expect an anti-clockwise arc. And using next generation sequencing, we can now take a snapshot of the reaction and find out what actually is happening. So each of these dots represents a product of the reaction, and the sort of size of the dots represents kind of how much there is. And you can see there is a nice clockwise arc here, kind of starting from here with the synthesis. But as you can see up here, there's other products. So what's happening? There is actually the first kind of, after the first kind of triplet ligation, the enzyme likes to connect to the second kind of dinucleotide and form a second arc. So there's two arcs kind of going to the full-length product. And you can see what we conclude from this. There's two independent initiations from two triplets and independently extending. Now let's look at a different, slightly longer, a 27-mer template. Now already just looking at the pattern, you can see this is very different. So this uses a very different pathway. You have this time quite a nice developed anti-clockwise arc, but you can see you have all these other kind of arcs here as well. And just took a long story short, if you try to reconstruct the pathway, you come up with at least four independent initiation events, both in the forward and the reverse direction. So what's really exciting is the system, as we ask it to replicate RNA, can now find its own way. It can initiate where it wants, fill in the gaps, and sort of find the path of lowest resistance to find to make the final product. Okay. Now, another really important aspect of RNA-catalyzed RNA replication is if we want to go to self-replication, this reaction must proceed with a certain fidelity, which is defined by the famous error threshold of Manfred Eigen, because if you do not operate above the error threshold, the majority of the sequences you generate will be basically inactive. You will have destroyed the phenotype, and there can be no inheritance and no evolution. Now, one way to enhance the fidelity of RNA replication, and obviously fidelity is a problem that is also occurring in biology, and one way to enhance the fidelity is to not just rely on the base pairing, but to actually read out the hydrogen bond donor and acceptor patterns in the minor groove. And this is done both by DNA polymerases and in the decoding center of the ribosome. And in fact, my colleague Venki Ramakrishnan sort of speculated on the basis of the structure of the ribosome decoding center that really the primitive replicase, the RNA replicase that we're looking for, if it was able to read out minor groove hydrogen bond donor patterns, its fidelity would be much improved, and which would potentially allow much more accurate replication and more complex systems to evolve. So we thought we should really try to select for fidelity. And how do you do this? So we started by feeding, the main source of error of these RNA replication reactions are GU wobble pairs. So we presented the ribozyme with two triplets, the correct one, and a sort of a terminated triplet with the wobble pair. So the terminated triplet cannot be extended. So if the polymerase incorporates it wrongly, it cannot synthesize to the edge of, to the end of the strand. And as you can see during selection, initially the wobble triplets actually incorporated slightly better, but as we keep going, eventually its incorporation disappears. And at the end of this replication experiment, if you look at the fidelity of the triplet polymerase, so this is for the first, the second, and the third position of the triplet, you can see initially the fidelity overall wasn't very bad, and it was particularly bad for the third base of the triplet, but after evolution, both the second and the triplet have much improved, although there's still wobble pairing in the first base triplet, but the overall fidelity has now improved to about 97%. Now, to find out if this really is based on hydrogen bond pairing, I'm going to show you some cool experiments done by Aditya Raghuram, who is I think, a group leader now here at the Broad, and was in Cambridge briefly kind of for a summer internship and did some cool experiments by testing essentially if we could prove kind of that the minor groove hydrogen bond donor acceptors were read out by replacing the uridine, where the keto group is like often used as a hydrogen bond donor acceptor, replacing this with 2-thiou, which can't form a hydrogen bond, and also replacing the 2' hydroxyl of RNA with a fluoro group, which again kind of abolishes hydrogen bonding. And you can then measure the impact on fidelity if you have this modification present or not. And the experiments he did sort of clearly showed that at the third triplet position, the minor groove is definitively engaged, because if you, for example, replace uridine with 2-thiouridine, the fidelity drops back to the baseline, while at the first position, there is very little effect. Now, how does this work? Now, unfortunately, the cryo-EM structure that we have only is of the APO enzyme. So we don't know exactly how the triplet polymerase ribozyme engages the primer template duplex and the incoming triplet. But we can build a model based on the structure of the class 1 ligase ribozyme. And if you do that, if you fit the primer template into our structure, you can see in red the active site and the catalytic site in green, and the A minor kind of interaction motif in red. And in blue, you can see this P10 loop, which emerges during the fidelity selection, which is sort of poised to interact with the primer template duplex. And in more detail, you can see it is sort of perfectly positioned to actually hydrogen bond to the incoming triplet here in red. And this is the P10 loop. And this, I think, what gives us the high fidelity in the forward reaction, because in the reverse reaction, the P10 loop is in the wrong place and is positioned over the primer and can't contact the incoming triplet here in red. And this may explain kind of why the reverse reaction has a much lower fidelity back to baseline. Okay, so there's another way in which triplets kind of seem to influence fidelity. And this is completely unexpected and is based on the sort of systems level properties of the triplet pools. So the first hint we got from that is when we looked at the fidelity of two triplets. So this is pretty much the worst case scenario. the ribozyme really discriminates those very, very poorly, that in 30% of the cases, the wobble pair triplet is actually incorporated. But before I do this, just to notice, if we have a pool of 64 triplets, for each triplets, there is also its perfect anti-triplet in the pool. Now, what we did is if you provide those two triplets, their anti-triplets, the fidelity actually improves by over 3-fold. Now, this is totally unexpected because those triplets have nothing to bind to in the template. The template just codes for ACC incorporation. Now, if you use the same template and you give it all 64 triplets, another weird effect pops up, because if you increase the triplet concentration, again, normally would expect with increasing triplet concentration for the fidelity to drop, but in fact, the fidelity increases. Let's look at another example. Here, the fidelity phenotype isn't so bad, but again, if you provide the anti-triplet, it gets almost 4 times better. On the same template, you use the NNN pools, again, the fidelity increases. So what's going on? Now, if you look at basically all the triplets, and you look where the fidelity effect is the most pronounced, you can see only above 50% GC is where you see a significant shift. And you can look at this here, where you look at the fidelity effect at high versus low concentration of NNN triplets, and all the AU-rich triplets behave as you would expect. At higher concentration, the fidelity drops. But for all the GC-rich triplets, the fidelity increases at higher concentration. So what's going on? Now, we think kind of because this KD range is exactly in the sort of range that has previously been shown to promote dimer formation among tRNAs between codon and anticodons, I think our hypothesis is that these anti-triplet interactions in the pool of triplets, they occur selectively between the GC-rich triplets. And so what really happens is the pool sort of normalizes these interaction networks, increase the fidelity by normalizing the triplet substrate pool against base pairing strength. All the strongly pairing GC-rich triplets form dimers, and the concentration of those free triplets drops as a function of their base pairing strength, while the more weakly interacting AU-rich triplets don't form dimers and their concentration is increased. So overall, this is the same effect as if you would deplete the most mispairing triplets selectively from the pool. Okay, so here's another RNA replication problem. Whenever you replicate RNA, the product is invariably an RNA duplex, and RNA duplexes are insanely stable. And these double-stranded RNAs are basically, they cannot be replicated by the RNA polymerase ribozyme. They just are not substrates. And I just show you this here. Two strands, this is the plus strand, this is the minus strand. You can copy them happily. But you give it the duplex, you preform a duplex of the plus and the minus strand. There is no RNA synthesis whatsoever. And this is known as the strand separation problem. So clearly, we need to unzip the strands. So let's see how we can do this. Now, as I told you before, RNA doesn't like to be heated up, and particularly not in the presence of magnesium, which we need for ribosome activity. So how can we dissociate those super stable duplexes? Now, one idea came up. My colleague John Sutherland came up with this idea to actually use acid. RNA is surprisingly stable at acidic pH, and the base pairs begin to protonate around pH 4, and obviously this destabilizes the interaction. Unfortunately, to really kind of separate the sort of duplexes we like to replicate, especially in the presence of magnesium, which further stabilizes them, you have to go back down to maybe pH like 2.5 to really get good dissociation. And you also have to elevate the temperature. So RNA duplexes can be dissociated, but it's hard. And you need to use conditions that are not compatible with ribozyme activity. So you then need to neutralize again to do synthesis. But what if you do that? You see the duplex can't be synthesized. Now we melt the duplex with acid and neutralize and add the ribozyme. Now you can do it. Now you can synthesize both strands with somewhat reduced yield, but it can be done. So double-stranded RNA can be dissociated by acid pH and high temperatures, and the dissociated strands can be replicated. So now we have a way to actually do some sort of ribozyme PCR. Now we can go through multiple cycles of replication. And I should say that if you don't replicate it, the strands rapidly zip up together again. And we can measure that. The half-life is about 3 minutes. In about 3 minutes, all the RNA has reformed the duplex. And that is unfortunately too fast for the ribozyme to copy it, because the ribozyme is really not a fast enzyme. But again, triplets come to the rescue, because if you dissociate the strands and then add the triplets before you neutralize, the strands can no longer anneal. So the triplets actually attenuate, they kinetically and thermodynamically stabilize the dissociated state, so to enable replication. And this just shows you sort of a sketchy delta G diagram where you dissociate the single-stranded state is a sort of high-energy state which wants to go back to a low-energy state, which is the duplex. But because the triplets bind to the dissociated strands, this A becomes destabilizes it thermodynamically, and because you now have to strip all the triplets off before you can zip back together, it also slows it down kinetically. So this really stabilizes the single-stranded state and slows down strand annealing. And this does not work with the mononucleotide triphosphates. So when we do this with the old RNA polymerase ribozyme and the mononucleotide triphosphates, you can't copy the duplex, even if you have dissociated it with acid. Okay, so now we've got a scheme that we need to do. We need to dissociate the strands with acid, neutralize, add the triplets and the ribozyme, and replicate and go through these cycles. But there is a problem. For dissociation, we need low pH, high temperatures, and low concentrations of everything, in particularly RNA. For synthesis, we need actually mildly basic pH, we need low temperatures, and obviously we need high concentrations, ideally, of everything. So these are sort of really opposing conditions. But again, something I told you before earlier comes to the rescue, because we can now kind of have this tremendous take advantage of the tremendous concentration effect of ice, and also to bring the temperature down. So if we now combine hot temperatures and low pH with freezing and high pH, we should be able to, by coupling freeze-thaw cycles and pH cycles, we should be able to decouple the strand separation and the synthesis steps. So this is just a little cartoon which shows you how this should be working. You start with a duplex, you move it to pH 3, heat it up, they come apart. You add the triplets, you move it to pH 9, and you freeze, and add the ribozyme, and now it copies it. We move back to pH 3, they come apart, and so on and so on. And you should be able to get exponential RNA replication. And I would say this cartoon is a little bit optimistic because it shows a doubling. In fact, it's not so good. You only probably double in the best case scenario every 5 cycles. But you can now use this kind of, if you want, acid-free cycle PCR kind of to really kind of replicate and amplify double-stranded RNAs. Now, what's more interesting than this is what happens when you feed the system not a defined sequence with primers, but actually a random sequence library and a random sequence template with primers, with defined primers attached. Now, I think this is really weak, but this is the original length template, and you can see in the first 5 cycles, this is sort of synthesized. You can see a little bit of the full length template, but as we keep going through more and more cycles, the system sort of shifts to a different sequence population, and you can see these strong product bands emerging, which are truncated from the full length. And these actually grow exponentially over many cycles. And these sequences, when you analyze it, it's quite a diverse pool. But these sequences have now been selected by the ribozyme for being nice to copy, for what I would call replicability. So the system itself actually selects for highly replicable RNA sequences. They emerge spontaneously from a random pool and then amplify exponentially upon continued RNA replication. And you can pick out some sequences from this of the different lengths, and you can see they nicely grow over 5 cycles and keep amplifying. Now, I've told you before about primer-free replication. Now, what happens if you do this without any primers? So in this experiment, all we feed into the system is an N20 seed sequence, all 64 triplets, the ribozyme, and we just let it go. As you can see, initially it copies the N20 seed, but then very rapidly a sort of ladder kind of products appears, which grows in intensity as we keep going. Now, what is really interesting is we also do a control where we don't add the N20 seed sequence, which initially shows a sort of very weak pattern, but then this sort of banding pattern appears, and then the sequence the reactions which had no template input at all, they actually begin to overtake the ones which had an RNA template. So what goes on? So we analyze these pools by next generation sequencing, and what you find is that actually a large number of these sequences are actually homologous to the ribozyme. So this is now in the pool which had not had a template sequence added. So essentially, in the absence of a template, what the ribozyme does, it uses itself as a template. So it first kind of synthesizes pieces of the minor strand, and then these eventually become used later on as templates to synthesize the plus strand. So these are all sort of small fragments. I would say these are not contiguous sequences, but this is all sort of small pieces of the ribozyme that are being replicated and churned. So really, the primer-free RNA replication in the absence of an N20 seed yields an expanding population of ribozyme minus and plus strand fragments which are being continuously replicated. But the polymerase also does something else. You can see this sort of red section which increases as a replication goes on. There was no template added to this reaction, so this is not homologous to the ribosome. This clearly must represent the de novo generation of replicating RNA sequences. So the polymerase sort of dreams of RNA sequences and begins to sort of doodle and create new sequences which then get replicated. And we think this is probably through the addition of these GC-rich kind of triplet pairs. Now, when you sequence specifically these sequences, you also notice something interesting because they have a highly biased sort of triplet code. And if you superimpose that on the genetic code, what is quite striking is that this matches very well with the so-called family box codons, which are shown here in gray, which some people think is representing the primordial genetic code. Now, this may simply be, one has to be aware of, if you want, confirmation bias, because it's likely that this is at least partially just a function of thermodynamics, because the family box codon triplets also tend to be the triplets which are bound most stably kind of to the template. But what certainly is true is that this type of open-ended and unbiased RNA replication generates sequences which are inherently biased to be translatable, because they use codons which may have been present and certainly contain no stops. Okay, so everything I've told you so far was sort of based on this ribozyme, which I've shown you the cryo-EM earlier on. As I said, it's a mutualistic heterodimer with a inactive subunit in blue, and here the catalytic subunit. It's an advanced phenotype, but it's complex. It's really kind of quite a beast. And it's 288 nucleotides long, so this is going to be challenging kind of for self-replication. If for each cycle we have to make nearly 300 nucleotides. And also, if you think about the idea that such an RNA needs to emerge spontaneously from a pool of random sequences, the probability is vanishingly small. So here comes another paradox. You need complexity for function, but actually for emergence, what you want is simplicity. And we sort of avoided this issue for a long time, but eventually we decided we had to address it. And we tried to start again and do a de novo selection for triplet polymerase activity, and this time with just very small kind of RNA libraries, selecting for triplet incorporation. And what we got is these sort of 3 small RNA motifs, which emerged really from a very small kind of random sequence library and show some polymerase activity. We picked one of them and evolved it further. It's not an enzyme with an amazing catalytic turnover, but it actually has very robust polymerase activity, and particularly if you compare it to the original ribozyme. And this is all now encoded in this really small domain of just about 50 nucleotides. And Eduardo, the postdoc working on that, kind of like making fun of sort of British understanding, sort of like called this the quite tiny triplet polymerase ribozyme. So from now on, I'll refer to this as the QT ribozyme. And yeah, so this a small 50 nucleotide RNA motif with robust polymerase activity. Okay. Amazingly, this can be truncated further, although you start to pay an increasing price in polymerase activity. But the sort of QT45 still has very acceptable polymerase activity. And you can now use this to make other ribozymes. So this just shows you the synthesis of a mini hammerhead ribozyme, both with the sort of defined triplet substrates and with a sort of random, all 64 random substrates. You can cut out that band on the gel, isolate it, and you can show that these ribozyme-made hammerhead ribozymes are actually active. They can cleave an RNA substrate. And they are synthesized with, really, it's not such a good fidelity under those conditions, but actually they're very matching fidelities between the QT and the more complex 5TU polymerase ribozyme. So really, the QT ribozyme is able to synthesize an active mini hammerhead ribozyme, and it has a sort of general polymerase activity on a number of sequences. I haven't shown you those, kind of always with the same fidelity of about 94%. Okay, so now we've got a polymerase ribozyme, which is small, which actually has robust polymerase activity. Now the question is now, can it make itself? And really, those are the 2 component reactions of self-replication. You need the minus strand synthesis, where the plus strand, the folded QT plus strand, uses itself, another plus strand, as a template to make another minus strand. That's the minus strand synthesis. And then for the plus strand synthesis, you need the plus strand, the folded QT, to use its complementary strand, the minus strand, to template the synthesis of another plus strand copy. So I'm going to first talk about the minus strand synthesis. Now, if you think about it, here's another paradoxical requirement. This is now, you need the QT plus strand to use itself as a template. So in this reaction, the plus strand needs to be both ribozyme and template at the same time. So how can you affect this, that it is both a string and a thing again? Is it both folded and unfolded at the same time? Again, the triplets come to the rescue because, as I told you before, the triplets can invade secondary structure. So you can have this state where it is both unfolded and folded as part of an RNA conformational ensemble, which is essentially affected by triplet concentration. So at low triplet concentration, you can see you have 2 copies of the plus strand. They're both folded. Nothing happens. But as you increase the triplet concentration, an increasing population of plus strands unfolds and becomes coated by the triplets, and then can be used as a template to synthesize the minus strand. And if you go too high with the triplets, eventually all the strands are unfolded and there's no more activity. And you can see this on this gel. This is a sort of polymerase ribozyme activity as a function of triplet concentration. You can see there's quite a sharp transition where all the strands begin to unfold and where you lose all activity. So really the triplet concentration allows you to control the conformational ensemble, to shift the equilibrium from ribozyme active to template inactive conformation. And if you're at the right position, you have a sort of a population which contains both folded and unfolded plus strand. And then you can synthesize a copy of the minus strand. You can see this here. This is the full length band. And again, you can sequence this. And again, we get about 94% fidelity. So the first component reaction of self-replication can be preceded. So the QT45 ribozyme can synthesize a full-length minus strand copy from a plus strand template using random substrates. Now we come to the plus strand synthesis. Now here's the same problem. Now the ground state of incubating a plus and a minus strand together, as I first told you before, is an RNA duplex. This is incredibly stable. So we need to dissociate that. And as I told you before, the triplets allow us to stabilize the dissociated state. And at the right sort of concentration, you have in the dissociated state, you have some minus strands coated with triplets, obviously also some plus strands coated with triplets, but also some plus strands folded that can then use the minus strand as a substrate to synthesize more plus strand. And again, if you go too high, everything gets coated by triplets and there is no activity. But unfortunately, there is another equilibrium going on. And that is what happens is if the polymerase ribozyme, the plus strand concentration here is just a little bit too high, the ribozyme can compete effectively with the triplets to strip the triplets off the minus strand, and everything kind of goes back to the duplex, which is inactive and kills the reaction. And this is sort of shown here, that by increasing the plus strand concentration, you get less and less product formed. And this sort of shows this equilibrium. As the plus strand concentration increases, you more and more effectively compete with the triplets until you basically fold just plus strand and the duplex. So we thought that one way to fix this would be, instead of using triplets, we need to use a substrate, just in this case, that competes more effectively with the plus strand for binding to the minus strand. And this is particularly the case for an AU-rich kind of apical loop in the ribozyme that where the triplets kind of do not bind very stably to the template, and therefore this is single-stranded and sort of prone to invasion by the plus strand. And yes, if we add a hexamer that sort of blocks off this vulnerable site, we now can get full-length plus strand synthesis. So again, the triplet concentration allows controls of the strand conformations, and the hexamer substrate, complementary to the apical AU-rich loop, reduces the duplex formation and now enables the QT45 plus strand synthesis. This is kind of coming to the end of my talk. Just to show you here, it's a weak band, but we can also make the plus strand synthesis. So really the QT can complete both of the component reactions of self-replication. And we're sort of excited kind of to take this to the next step and go hopefully to a fully kind of self-replicating system that can begin to evolve. So as a summary, kind of like I've been trying to tell you kind of like how triplets really have some very favorable properties to enable RNA-catalyzed RNA replication, to promote it by opening up template secondary structures for replication, by attenuating strand reannealing after duplex melting. So it really solves the strand separation problem by what I would call substrate-assisted strand separation. It enables these non-canonical RNA replication modes. I've shown you the reverse and the primer-free, but you may appreciate that with triplets, you also potentially have 3 different registers in which you can synthesize RNA. It improves replication fidelity by these sort of systems-level triplet, anti-triplet interactions in the pool. And it enables this open-ended primer-free RNA replication and de novo generation of RNA sequence pools. And then the small QT ribozyme really now achieves the complex RNA polymerase ribozyme phenotype in a much smaller motif, just 45 nucleotides, and is able to, of general RNA templated RNA synthesis with good fidelity at 94%, and is able to complete the 2 reactions needed for self-replication. And really, for the QT, we're just at the beginning because we've only done about 18 rounds of evolution, and this is exactly where Dave Bartel's ribozyme was. It's called R18. It was at this stage right at the beginning of the talk, so this takes us back. And finally, I'd like to end with a sort of a plea to take ice seriously as a potential prebiotic primordial medium where RNA could have made more sense. Because really, if we think of our solar system as potentially a typical planetary system in the cosmos, of which we now know there are many, it's very noticeable that while liquid water is only found in any quantity on one planet, ice is abundant, ice is everywhere. There's whole celestial bodies in the outer solar system made from water ice, which would have substantial eutectic phase kind of like zones. Okay, and I'd like to end by thanking the amazing people kind of who've done this work. Pretty much everything I showed to you was done by 2 amazing postdocs, Jamie Atwater, who's now left the lab and running his own group at University College London, and Eduardo Gianni, who's still with us in Cambridge, and also with help from Sam Kwok, Kevin Goichi, and Bryce Clifton in the lab, and actually also some very, very able LMB interns which contributed to this, like Joe Curran, Aditya Raghuram, and Teresa, who are both here. And I'd like to thank Ebbe Anderson at Aarhus kind of for some great collaboration on the cryo-EM. These bodies for giving us money, and you for listening. Thank you.
So thanks so much for the amazing talk. Any questions? We prefer to give chance to students and postdocs to ask questions first. So any questions? You can either work to the microphone or I can just hand this to you.
Are you guys interested at all at looking at quadruplet substrate codons for?
We did right at the beginning, and I mentioned it briefly, but it probably went over it too quickly. So what happens is, when you look at the sort of progression from dinucleotides, tri to quadruplets, etc., as you go longer, they get incorporated more and more effectively because they bind to the template more effectively. But when you then measure the fidelity, there's a very sharp drop as you go from triplets to quadruplets. With quadruplets, basically one of the base pairs no longer matters. So that really kills the fidelity. So I think triplets is a sort of sweet spot at ambient temperatures. And yeah.
There are various clinical situations in which patients are treated with protein, giving them injections of proteins in order to supplement whatever is deficient. And sometimes they become resistant to that because they make antibodies to the protein.
Right.
Have you considered trying to make an RNA that would have the specific activity of a given protein that's used clinically, where that might be a substitute for the kind of situation that I just articulated?
Yeah, I mean, I think pure RNA is probably wouldn't be a great clinical substitute because it's not stable in the body, kind of in the serum, gets rapidly degraded. Modified RNAs kind of can form into 3-dimensional structures and potentially act as ligands like antibodies. They can also be enzymes for a sort of limited set of reactions. But I think in most cases what happens is that So for example, aptamers have been explored with chemical modifications in clinical trials to potentially replace antibodies, but the antibodies just tend to win out, sort of tend to be better. So I think it would very much depend on the protein you're trying to replace.
Actually, I have a lot of questions. Maybe I'll try to ask 3 to begin with, if no one has questions. So the first one is regarding the evolution of the ribozyme. Do you think the template-free replication is actually its own in vitro evolution system, which means if you begin with a RNA polymerase that's relatively error-prone, and then if it's replicating itself, then whoever most catalytically potent will be the final products?
Yeah, no, I mean, this is what we would like to do. We would really like to begin the self-replication process. So, you know, the system should become self-optimizing, and this has been literally proposed. You know, as you acquire mutations, you know, you select for better and better self-replication.
Yeah, they should emergence as the most efficient and the fatal system.
That would allow you emergence, but I think the problem with the fragmentary replication is that you keep losing the connection between the pieces. So even if if you replicate in fragments and then assemble them and then select for function, and then you would go back to the open end, to the randomly primed replication, you then break the connection again between the mutations. So I think the small ribozyme, we think, is the way to go.
Cool. And my second question is just generally, if this is indeed true, that's how things begin on Earth, do you imagine there will be another ribozyme synthesizing the triplet?
Well, I mean, I'm not sure I fully understood the question. We had 3 motifs which had triplet polymerase ribozyme activity. So there's clearly loads. That was a small RNA library. So I think if we keep selecting, I think emergence of these small motifs is really easy because they're small, they're simple, and there's probably loads of them.
I see. And then regarding the potential possibility of the evolution of ribosome in the end, do you imagine there could be some co-evolution of ribozyme and peptides to extend the self-replicate, now actually synthesizing some peptide products? Like, I feel the triplet is already the precursor of tRNA.
Yeah,
I
think, I mean, in a way, the evolution of translation is almost more mysterious than, you know, the sort of original kind of origin of life, because you need at least 3 different subsystems to sort of come together to give you translation. You need a reader, like the small ribosomal subunit that reads the sequence. You need a decoding adapter like the tRNAs, and you then need the peptidyl transferase. Now, some people have speculated that the S30 subunit of the ribosome, that's the old replicase that has become repurposed for translation. But I really don't know if, you know, I would like to speculate too much. But what is very interesting is that, you know, the ribosome, the genetic code, is a triplet code with 1 wobble position. And that's exactly what we have as well. With our triplets, we also end up with 1 wobble position. It's at the other side of the triplet, but it's the same sort of idea.
Yeah, I think it's quite amazing because they go from either direction, but then still they have this triplet, like the effects at the wobble position. So,
in fact, Jamie, I think, has some interest in, you know, building simple models of how translation might have evolved kind of in his lab. So, you know, you should ask him maybe in a few years' time.
Cool. Any more questions?
Sorry. Do we have more time? Okay, thank you. Great talk. Do you have any idea of how ribozymes can bias towards self-replication rather than replicating other RNA template?
Yeah. So what you're asking is, like, if you have the ribozyme in a soup of sequences, how does it recognize its kin? Yeah. Yeah, I mean, I think I haven't included this here, but I think the answer to that is the same as in biology, you need to compartmentalize. So you need to have some sort of, you need to sort of enclose the replication reaction into some sort of compartment, which means kind of that the ribozyme and its kin sort of stay together. And these type of compartments could be many things. They could be simple bilayer membranes, vesicles, and there's all kinds of pockets that you can have. In fact, I haven't shown you this, but if you set up the eutectic ICE phase in a particular way, instead of getting these channels of eutectic phase, you actually get little inclusions. So these could be, even the eutectic phase could be some sort of primitive, protocellular medium. And of course, you can hit pockets in sort of, if you mix kind of like, oil phases, kind of, and water type of emulsions. Some people are interested in aerosols, kind of, that you get, let's say, in ocean spray, which form like little droplets, and so on. So I think there's no shortage of enclosing compartmentalization modes that one could explore. But I think what we're sort of focused on getting the self-replication process to work. And I think once it's working, I think we'll definitively think about exactly that problem.
Hey there. I was curious if you could speak to any structural similarities you've seen between the smaller ribozymes and any larger ones, or between the smaller ones?
I don't think we really kind of see any, and I mean, what is interesting is we mapped the way the two ribozymes interact with the primer template, and they seem to contact exactly the same positions on the primer template. But when we try to look at the structure, I can show you a slide that I sort of have excluded from this. This is, when we sort of try to figure out what the ribozyme looks like by footprinting and sort of fitness landscape analysis. I mean, you can see it really kind of just looks like a hairpin, and we have some preliminary cryo-EM data which also suggests kind of it's just kind of, there's nothing that looks, like the other structure at all. It's just a hairpin structure. And we definitively, want to figure out how this thing does its job, because at the moment it's completely mysterious.
Thank you.
Any more questions? Okay, if not, thanks everyone for coming, and let's all thank Dr. Holliger's great talk. Okay.